Hey everyone, I hope the exam was good, and these are some questions from DADH that we have recalled from the NEET. So let's start with the first question. What amorous glands are primarily? Good evening, Nikesh. I hope exam was good for all of you. So let's start with the first question. And if you see any question that was not in there, please let me know. And if you find that I have missed any question, then also please let me know. We'll discuss those questions as well, right? So the first question is, what glands are primarily? And the options are mucous gland, serous gland, mixed mucous gland, and mixed serous gland. So I just had a couple of options, so I had to improvise on the options. Von Apner's glands, basically these are minor glands. Right, as you have studied in the class, these are minor salivary glands, and these are associated with circumvallate papillae, isn't it? And these are the only minor salivary glands that are pure serous, right? These are the only minor salivary glands that are pure serous. So I hope you all got this question correct. So let's go to the next question. The next question is, Merkel cells are seen in which layer of epithelium? So what are Merkel cells? Merkel cells, I hope you all remember that these are the non-keratinocytes, isn't it? These are the non-keratinocytes. And apart from Merkel cells, the other non-keratinocytes are lymphocytes, Langerhans cells, and melanocytes. So these melanocytes and Merkel cells, these are seen in the basal layer, isn't it? Merkel cell and melanocytes, these are seen in the basal layer, that is stratum basale. Whereas Langerhans cells and lymphocytes, those are suprabasal, or you can say they have variable presence, at least lymphocytes, right? So here the correct answer would be stratum basale. Is it clear? If you have any doubt, if you have any problem, do let me know, right? So second is, Merkel cells are seen in which layer of epithelium? They are seen in stratum basale. The next question was to identify a structure marked with the red circle in the given image. So as you can see, this is the image of a developing tooth germ, isn't it? It's a developing tooth germ, and the stage is cap stage, isn't it? In the cap stage, we see certain temporary structures, and this red dot is marking one such temporary structure. And as you can see, this temporary structure is close to the inner enamel epithelial layer, isn't it? So this is the red structure marked here. And what is this red structure? It is nothing but the enamel knot, isn't it? Enamel knot. Enamel knot is a group of cells that is seen associated with the inner enamel epithelial cells. From there, the temporary cells or the epithelial cells that extend towards the outer enamel epithelium, that is known as enamel cord, isn't it? Dental papillary cells are these cells which are the ectomesenchymal cells which are seen condensed here, isn't it? These are seen condensed here in the concavity of this cap stage, right? These are the dental papillary cells, and these are the ectomesenchymal cells. Whereas enamel niche is a temporary structure which is seen in the lateral lamina. It is nothing but a defect in the lateral lamina which contains again ectomesenchymal cells, right? So the structure that is marked here is enamel knot. So the next question is, mesiolingual developmental groove is present in. So this is from dental anatomy, and the mesiolingual groove is seen in the mandibular first premolar, isn't it? It is seen in the mandibular first premolar. So this is the mandibular first premolar occlusal view, and you can see that on the mesial side, on the mesial marginal ridge, towards the lingual side, a groove crosses over. Right? This is known as the mesiolingual developmental groove, and it is seen in the mandibular first premolar. Is it clear? Is there anything that you would like to ask? क्या अभी तक सब क्लियर है? आई होप सबके आंसर सही हुए होंगे, because these questions they look relatively easy. So which of the following is true regarding the following image? So this is a polarized image. This is a polarized image, isn't it? First of all, it is viewed under a polarized microscope. As you can see these colorful structures, isn't it? Polarized microscope. Also, it is a ground section. You can clearly see the enamel here, isn't it? So that's why we can say that this is a ground section, right? Because the enamel is lost in a decalcified section. So this is a ground section, and the arrow marked here, it represents the cemento enamel junction. And I hope you can all identify the type of cemento enamel junction here. It is not an overlap junction. Again, not an overlap junction. Rather, it is a, as you can see, the enamel and dentine, they have a butt joint, isn't it? So it is a butt joint. So CEJ butt joint in decalcified section. This again is incorrect. This is not a decalcified section. This is a ground section. So CEJ butt joint in a ground section, and it is viewed under polarized light, right? So let's go to the next question. As you can see here, this is the colorful tissue. That's why we came to the conclusion that it is viewed under polarized light. In normal transmitted light, it would have just appeared black and white, right? Which stage of amelogenesis is marked by figure 5 and 6? So figure 5 and 6. So basically this diagram has been taken from Ankit, and these represent different stages of the life cycle of ameloblast, isn't it? So when you start from the beginning, this is the inner enamel epithelial cell, then it elongates slightly in the organizing phase and it converts into pre-ameloblast. After that, just at the beginning of secretory stage, it converts into ameloblast. That is the third stage with the Tomes process. So this is a proper ameloblast. After secretory phase, it goes on into the maturation phase, and in the maturation phase, it will show two cell morphologies, a smooth ended and a ruffled ended. So 5 and 6 represents those smooth and ruffled ended ameloblasts. So basically, it represents the stage of maturation, isn't it? And 7th represents post ameloblast, or you can say this represents the reduced enamel epithelium. It represents the reduced enamel epithelium at the stage of REE. 7th represents at the age of, or at the stage of REE, when it converts into reduced enamel epithelium. So let's see if we have a maturative stage here. So option D is the correct answer, maturation stage, right? When the enamel is undergoing maturation. So let's go to the next question. Accessory cusp present in mandibular molar is. So we have seen the accessory cusp in the maxillary molar. In the maxillary molar, it is called cusp of Carabelli. So it is present in the maxillary molar. It is present in upper 6 and in upper E. But if at all accessory cusp is present in mandibular molar, what would it be called? It would be called protostylid. So protostylid is the accessory cusp that is present in mandibular molar, and usually this protostylid is present on mesiobuccal side of the lower 6. So that is the common location of protostylid. Then now there is another accessory cusp that is associated with lower first molar, and it is called tuberculum intermedium. Now this tuberculum intermedium is associated with the lingual surface, and it is found between, so let's say this is the first molar. Let's say this is the first molar, then this tuberculum intermedium would be found between the mesiolingual and distolingual cusps. So this is the tuberculum intermedium, right? If at all an accessory cusp is present, let's say this is the distal margin, right? And this is the mesial marginal ridge. If at all the accessory cusp is present on the distal marginal ridge, let's say this is a accessory cusp present on the distal marginal ridge, then what will it be called? Now, when present on the distal marginal ridge, it will be called tuberculum sextum, right? Then it is called tuberculum sextum. So based on what you have in the options, you can mark that. So either it is protostylid, then it is present on the mesiobuccal side, so it would be present somewhere here, right? It is present on the mesiobuccal side, then it is protostylid. If it is present between the two limbal cusps, then it is tuberculum intermedium. If it is present on the distal marginal ridge, then it is tuberculum sextum. So these are the three accessory cusps that may be associated with the mandibular first molar. right? Just remember protostylate, tuberculum intermedium and tuberculum sextum. So next question, based on the image shown below, estimate the age of the subject. So I am not sure whether this is the exact image which you had in the question, but I hope this resembles the image that we had in the question, right? So what we see here is that we have all the primary teeth present in the oral cavities. At this stage, we have all the primary teeth. None of them have exfoliated. Plus, what we can also see is that amongst the permanent, the first permanent molars have just erupted. And we know that the age of eruption of the first permanent molars is around 6 years, right? So we can safely say that this subject is somewhere around 6 years of age, isn't it? So let's take a look at the options. 4 plus minus 9 years, 5 plus minus 9, 6 plus minus 9, 7 plus minus 9 months, right? So obviously, since the first molars have erupted, the subject is somewhere around 6 years of age, and hence the correct answer here would be 6 plus minus 9 months of age. I hope this is the diagram that you had in the exam. Now, if there are some variations, we can adjust the age according to that. If, let's say, if the molars were still unerupted, right? If the first molars were still unerupted or were just about to erupt, and the incisors were showing similar resorption, then also we can say that the subject is somewhere around 6 plus minus 9 months, because they are about to erupt. So, obviously, either it is 6 years or maybe 6 months less or 6 months more. So even then option C would be correct answer if they are about to penetrate the oral cavity, right? So next question, Orland bodies are. So what are Orland bodies? So Orland bodies we have studied in oral epithelium, isn't it? They are also known as keratinosomes they are also known as keratohyalin granules, isn't it? And what do they secrete? They secrete loricrin and involucrin, isn't it? They are responsible for increasing impermeability of the oral mucosa, isn't it? They basically make a barrier against the permeability. So they secrete loricrin and involucrin, isn't it? So let's rule out the options one by one. First of all, as we can deduce from the description, they are secretory organelles. They are not resorptive organelles, isn't it? So can we easily rule out that they are modified lysosomes? Isn't it? We can easily rule out that they are modified lysosomes because these are secretory and not resorptive, isn't it? Second is mainly present in stratum basal. Again, that is incorrect because they are found in stratum granulosum. They start forming at the superficial end of the stratum spinosum and then they are seen in the stratum granulosum. So again, option B is incorrect. Option C, secrete alkaline phosphatase. Now they do not participate in mineralization, isn't it? So that's why alkaline phosphatase does not have anything to do with them. So option C is also ruled out. Option D, modified Golgi apparatus. Now they are derived from Golgi apparatus, and that's why, and also they have a secretory. They also have a secretory function. So we can say for that matter that they are modified Golgi apparatus, and hence option D here would be the correct answer. Is it clear? So Orland bodies are not modified lysosomes. They are modified Golgi apparatus. इसी बात से समझो कि lysosomes they have the job of resorption, whereas Orland bodies they have the job of secretion. So that is exactly opposite, isn't it? So this is the last question. The last question, this was about a power stroke, a muscle that was involved in power stroke. So maybe they asked the name. So the language of the question might be different. I don't know exactly. Which of the following is mainly responsible for power stroke or mastication? Either this was the language or which of the following is the power stroke muscle. So in either case, the answer would be same, right? Options are medial pterygoid, masseter, lateral pterygoid and temporalis. Now what is the power stroke? Power stroke is that moment when, see, during mastication, first the mandible is depressed and then it is elevated. Once the mandible is elevated and it comes in the first contact, right? There is a contact of the teeth with the food, isn't it? So let's say this is the working side and this is the food in between, and this is the mandible, isn't it? So once the mandible is elevated and it comes in the first contact, right? There is a contact of the teeth with the food, isn't it? So let's say this is the working side and this is the food in between, and this is the mandible, isn't it? So once the mandible is elevated and it comes in the first contact, right? There is a contact of the teeth with the food in between, and this is the This, let's say, is the mandible. So when there is first contact from here till the teeth come into the interocclusal contact, that is the power stroke, right? So from the first contact till the interocclusal contact, that is the power stroke. So this is the time when maximum force is applied. Now in this power stroke, three muscles are participating: medial pterygoid, masseter, and lateral pterygoid, right? Sorry, not lateral pterygoid, it is the posterior fibers of temporalis. It is the posterior fibers of temporalis. Lateral pterygoid has more of a supporting role. So these are the three muscles participating: medial pterygoid, masseter, and temporalis. To be specific, posterior fibers of temporalis and superficial fibers of masseter. So these are the three muscles that are participating in power stroke. Amongst these three, it is the medial pterygoid which has the major role. No, Junaid, they are not modified lysosomes. As I just said, they are derived. Metaconus is also not the accessory cusp. And first of all, Orlan bodies, as I just said, Orlan bodies are derived from Golgi apparatus. Also, they have the role of secretion, unlike lysosomes, which have a resorptive role. So their roles are also different. So we cannot call them modified lysosomes, right? They are resorptive organelles, lysosomes, whereas Orlan bodies, they are secretory organelles. So they are derived from Golgi apparatus, though in true sense, they should not be called as modification of either of these two. Right? In the true sense, they should not be called a modification of either of these two. They are individual, they have an individuality, they are individual organelles, isn't it? But still, if we want to compare it to something, we can compare it to Golgi apparatus and not lysosomes. As far as metacon is concerned, metacon is not an accessory cusp, right? So the answer would be protostyle. That is an accessory cusp. As I just told you, there are three types of accessory cusp which are associated with mandible: protostylate, mesobuccal side, then there is tuberculum intermedium and tuberculum sextum. So these are the three accessory cusps. So let's come back to the power stroke. So medial pterygoid, masseter, and temporalis, these are the three muscles which are associated with the power stroke. And lateral pterygoid has a stabilizing action. Right? It has a stabilizing action. So it stabilizes during the power stroke. Now medial pterygoid, it has a mediolateral orientation, right? It has a mediolateral orientation. Also, superior inferior and mediolateral, as you can see, it is oriented mediolaterally and superior inferiorly. The power stroke also has the same direction as the direction of medial pterygoid muscle, and that is why we say that the medial pterygoid is the primary muscle involved in power stroke, right? It is the primary muscle involved in power stroke. So और ज़्यादा अगर हम डिटेल में जाना चाहें तो medial pterygoid, masseter, temporalis, all of these three on the working side, right? All of these on the working side. So if you are chewing food on the right side, then on the right side these muscles will activate to stimulate the power stroke. Is it clear? And lateral pterygoid has a supportive role or a balancing role. It stabilizes the mandible during the power stroke. Is there any other question? And metacone, protocone, protoconoid, these are all names of the cusp from the developmental perspective, right? So they are not usually asked. Metaconoid, these are all the developmental names, right? So these are usually not asked. So these were the 10 questions that I had as far as DADH is concerned. If you have any other question, you can ask me. Do you have any other questions? So if you don't have any other questions, then I hope these were the only questions that came in the exam, and all the best for your NICT exam. So take care and do well in the exam.